Okay, so I'm gonna try to let's say broadcast a little bit what we are doing in terms of collaborative framework in general within the scope of our P20 and more broadly on urology research that happens at Columbia and especially in my lab. So what we're talking about is congenital anomalies of the kidney and urinary tract, and here is the a quick cartoon that shows you, but you guys all know about it, of course, that any alteration in a number, shape, and structure and function that is developmentally related to the kidney and urinary tract is under this umbrella of CAKUT, and as you can see here, color coded. They are very highly variable in terms of phenotype, but also in prevalence in the general population, really highlighting a very impressive clinical heterogeneity that is also reflected by the genetic heterogeneity underlying these traits. So, what are the challenges in basic and translational research for CAKUT? First of all, the high genetic heterogeneity and complex genetic architecture require large CAKUT cohorts and also multidisciplinary approaches in order to identify new causal genes. Once these studies are conducted, these usually yield to very large number of candidate genes and variants that they then require adequate functional modeling in vertebrates, in vitro system to establish causation to start with. And once the causation is established, then we need mechanistic study to understand the pathobiology underlying each CAKUT predisposing gene and really working up the mechanistic insights into these discoveries. And eventually we'll have to return to human and its genetic and in its own genetic and epigenetic context in order to really understand what are the consequences of this mutation when they happen in the patient specific context. And this is going to be critical for devising targeting strategies. So our collaborating, let's say, collaborative framework really cover these points, and I'm going to try to show you a little bit of these examples of what we are doing at Columbia and how this can be brought into the more and more in the caribou interaction core. So everything starts from patient recruitment. And so just here to not to brag, but this is to broadcast a couple of papers, more recent from our group, and mostly because I often people make fun of me because how many authors do you need really to write a paper? Indeed, we have 50 to 100 authors per paper on average in our projects, and I would say that there's never enough authors. So each of these individual investigators has contributed significantly, and is demonstrated also by the large number of co-first and co-last authors in every one of these papers, because we really need the expertise of multiple investigators to complete the studies. So everything starts from patient recruitment, and here is a quick map of the worldwide sites that they contribute samples for human genetic research to our center. This is zoom in into Europe, and this resulted up to date in over 7,000 individuals, independent individuals recruited with CAKUT, plus additional several thousand of family members. And this, if we zoom in, just in Italy we have 31 divisions across 3 specialties: urology, nephrology, and pediatrics in 18 different provinces that they constantly and continuously recruit patients from our studies. But this is not a unidirectional type of work. We are actually returning genetic results. We are helping them and fostering clinical and translational research on site. As a result, they obtain already over 5 grants from the National Institute of Health in Italy and from the Teleton Foundation for supporting studies on the benign urology across Italy. And in the United States, of course, we have collaborative networks for patient recruitment. We established the DONAT, the development of the urinary tract, which is a clinical study which is led by myself, Patricia Weng at UCLA, and also Michelle Raoul. And this is supported by the Pediatric Nephrology Research Consortium in order to recruit patients with the CAKUT across multiple institutions in North America, including Canada as well. But we also have collaboration established for more basic science. And here is an example. I show you the work that we do with Erica Davis, formerly at Duke University at the time with Nico Katsanis, and now in Chicago at Lurie Children's Hospital, in which we develop models in order to use zebrafish as an intermediate tool to characterize and prioritize CAKUT genes. And I'll give you some example of how this model works. So we combine these cohorts. Here in this case, we have 3,000 cases with CAKUT and compared to 21,000 plus controls across the phenotypic spectrum from upper to lower urinary tract, and we conduct large-scale genomic studies in order to identify the genetic determinants. One discovery that we made pretty rapidly was that a large fraction, or like a significant fraction of patients with a renal hypoplasia, dysplasia, or agenesis, they carry microdeletion at chromosome 22q11.2, which is the DiGeorge locus. This was kind of known in the literature that about 30% of patients with DiGeorge, they have CAKUT, but what was striking is that our deletions, they cluster in the distal portion of the locus, and the parsimonious explanations that one of these genes, when mutated, would cause CAKUT independently. So there are still 9 genes. So how do we go from 9 genes to the single gene? And this is where the zebrafish came handy. And that's how we start modeling. So this comes from a more recent review. So while we all know that the mouse replicates pretty well the development of the kidney and urinary tract of humans, representing all the critical structures and segments in the nephron, the zebrafish, yes, has some conserved elements, and we can predict that perhaps upper urinary tract, they are more easy to model and interpret in the zebrafish, while the lower urinary tract perhaps less. But nevertheless, there is a significant conservation in the proximal and distal tubules. So we decided to try to develop readouts for CAKUT in zebrafish in collaboration with Erica Davis for both upper and urinary tract. So one that works pretty well for the upper urinary tract, we used the pronephric convolution as a readout for upper urinary tract malformation. So when you knock out a gene that is critical for the development of the proximal pronephric tubules, you see a simplification. This actually is the example of CRKL. This is the gene that we found in the chromosome 22q microdeletion. But we also try to develop more complex and less definitely accurate, but develop tools for interpreting lower urinary tract malformation. And here I'll show you the phenotype of the cloaca. And this is a normal cloaca when this is a gene that we studied, and you see that the cloaca is definitely abnormal in this model. So we develop readouts for upper urinary tract tubulogenesis and for lower urinary tract obstruction. Always keeping in mind that this is by far not the most perfect model, but definitely adds in speed and the ability to model multiple genes and perhaps even interaction between multiple genes that is not feasible in mouse model. But eventually you can go, we have to go back to the mouse, and this is actually what the genetic discovery, the characterization by zebrafish brought to then the analysis of CRKL for the DiGeorge locus syndrome. And you must have seen the poster presented by Jerry Martino from my group. We obtained a mouse construct from Akira Yamamoto in Chicago and Bernice Moore and Einstein. And Jerry nicely shows that the different CRKL isoforms are critical in and their balance is critical in the phenotypes of CAKUT observed in DiGeorge patients, by which absence of one or both isoforms can lead to a hyperbranching or hypobranching phenotype. And by then using transcriptomic analysis and the convolution to a pseudo single cell RNA sequencing, we can actually get insight into the mechanistics of this disease by observing that manipulation of CRKL leads to a clotto-dependent FGF signaling alteration that guides S-shaped bodies cells proliferation.
You've got about 1 minute left.
Okay, so then I'll skip the second one, in which we go straight to a mouse. But so what happens? So we can actually develop all these mechanistic insight. Then one of our main goal to the P20 was to go back to humans. So study of CAKUT fetal tissue to conduct multi-omic studies in the specific patient context. So our goal and that we are doing is trio-based genetic studies in which unaffected parents they have an unfortunate termination of pregnancy because of very severe disease, CAKUT. We sample tissue from these fetuses. We sample multiple parts of the kidney in the urinary tract to conduct single cell and single nuclear RNA sequencing, as well as ATAC-seq and multi-omic studies. These, of course, has some impediments to this approach. First of all, legislation has hit while we were doing these studies, restricting the use of human fetal tissue and the COVID-19 pandemics. So what we've done, we have developed alternative strategies. So we start using pluripotent stem cells and organoids as surrogates of human fetal tissue. And that's what we are doing now until we will resume the collection of human fetal tissue. And we always pair it with our mouse models. So we collect cells from patients, peripheral blood cells, and then we generate organoids that we transplant under mouse capsule to study the disease in a patient-derived context. So I hope in this very short time I give you a flavor of what is our P20 was and is about. And with this, I'll go with the acknowledgement, worldwide acknowledgement, patients and family member, my lab. And I also want to just stress that we are very dedicated to urology research. Junta Oke, postdoc in my lab, is the recipient of the this year American Urology Association Research Fellowship. Great Whitmore, you guys are getting to know him more and more. He's a medical student who's going to go into urology. Former trainees in my lab are Alejandro Perez and Jonathan Dia, who are now urology